In the previous class, I have mentioned that the light chain kappa and delta and the heavy chain consist of variable region, diversity, joining region. In case of light chain kappa, the variable gene segment can be of 34 to 38 and there is no diversity region. The joining region can be 5. In case of lambda, the variable region can be 29 to 33. There is no diversity region, D segment. Joining region can be 4 or 5. And in case of heavy chain, the variable region can be 38 to 46. There is diversity region 23, joining region 6, and all these light chain and heavy chain consist of constant region. So in case of kappa, the constant region is 1, for lambda 4 to 5, and for heavy it is 9. So these variable, diversity and joining region gene segments are present in many numbers in case of kappa, lambda, light chain and heavy chain. So this diverse number of gene segments actually helps in the combinatorial diversity to generate more diverse set of antigen receptors, or in case of the antibody production. This variable region and joining region in case of light chain combine to form the variable light region of the light chain. But in case of heavy chain, the V, D and J segments recombine. At first D and J segment recombine, then the V and DJ recombine to form the variable heavy region, as already I have mentioned in my previous class. Next is recombination signal sequence. This is 23 base pair spacer region and this is 12 base pair spacer region, but the heptamer and the nonamer are consist of 7 nucleotides and this consist of 9 nucleotides are very conserved. Heptamer and nonamer are very conserved, but this spacer region is variable. This is called 23 RSS and this is 12 RSS. You can see in this case of lambda chain, the V region of the lambda consists of 23 RSS, but now the J region must contain 12 RSS. And in case of kappa chain, if the V region or V gene segment consists of 12 RSS, the J segment must contain 23 RSS. And in case of heavy chain, the V chain if consists of 23, then the D should contain 12 and 12, and the J segment have 23 RSS, recombination signal sequence. Now this sequence, this gap sequence can be deleted out so that the two region V and joining region can recombine, and it can be actually come close to each other by inversion method. In this case, the signal sequence, receptors recombination signal sequence do not get deleted, but it actually exists in the chromosome, but this V and J segment comes close to each other in both cases. So deletion and inversion, by this both mechanism, the gene segment can become close to each other. Now the molecules that are involved in VDJ recombinations. RAC1 means recombination activating gene 1 and recombination activating gene 2 actually perform its first function in case of VDJ recombinations. These are the two enzymes that actually cleaved the RSS, recombination signal sequence. The next function is performed by Ku70 and Ku80. This is actually, these all proteins, just these RAC1 and RAC2 are in the, and the TDT are actually very lymphoid specific. Only the lymphocytes express RAC1 and RAC2 and TDT, these three enzymes, but all these Ku70, Ku80 and these enzymes are actually present in all cells because they are the components of non-homologous end joining, a recombination type of DNA recombination. So Ku70 and Ku80, these two proteins actually stabilize the DNA. DNA PK is actually an enzyme, protein kinase, that actually recruit Artemis, another enzyme, and actually phosphorylate the Artemis to activate the Artemis. Then Artemis actually clip the hairpin loop. TDT means terminal deoxynucleotidyl transferase. This enzyme has a very important role in VDJ recombination. XRCC4, Sarnunas and DNA ligase 4 works together to actually ligate the joint segment or the two gene segment. The mechanism of VDJ recombinations. If this V segment and J segment actually starts to recombine, first the RAG1 and RAG2 bind with the signal sequence. Then a synapsis occurs between these two segments like V and J segment. This RAG1 and RAG2 cleave the RSS sequence. The result is one is called coding joint where the V and J segment are present, and the generation of signal joints where the 23 RSS and the 12 RSS are present. Ku70 and Ku80 actually binds with the DNA ends, and you can see here the loop that is formed during the cleavage of RAG1 and RAG2. You can see the Ku80 and Ku70 proteins actually attach with the DNA segment, and this is the loop. Now DNA PK and Artemis, DNA PK actually recruit Artemis and actually is phosphorylate the Artemis. Then the Artemis actually cleaves the hairpin loop. You can see this hairpin loop is now open. Okay. When the loop is open, terminal deoxynucleotide transferase enzyme actually add non-template nucleotide. Non-template means this enzyme do not require any templates or parental template strand. Then DNA ligase 4, XRCC4 and Sarnunas proteins ligate the two gene segment like V and J gene segment. Now if this is a RSS sequence that present in the D region and this is the RSS sequence of the J region, they come close to each other, and RAG complex RAG1 and RAG2 generates hairpin loop. So how they generate? Because they cleave this RSS sequence, and now this TCAG is present in D sequence and this TAAT is present in the J sequence. What happens? This DNA PK and Artemis actually cleave, especially Artemis enzyme actually cleave this portion and in these portions, so that the loop becomes open. This loop, TCAG loop, becomes open. But one thing happens in this case, that is when the loop is open, it actually forms a palindromic sequence. If you see this, you can find that TCGA will produce TCGA alternatively. So this is called palindromic sequence. After palindromic sequence formation, TDT actually add N nucleotides. The palindromic sequence is called P-nucleotides, and the TDT formed N nucleotides. N means non-template nucleotides. Non-template means this enzyme do not require any template to form a complementary DNA strand. As we all know that to form a complementary DNA strand, parental DNA strand is important. Okay, but in this case, this TDT means terminal deoxynucleotide transferase do not require any template strand, and it can actually add abruptly any 4 nucleotides. So this actually create a high diversity, because what happens during replication, DNA polymerase actually add only the complementary nucleotide that depends on the parental strand. For this addition of this non-template nucleotide, this region that formed by the TDT actually is the main highest diversity region. Then the pairing of the strands, if any pair or any nucleotides do not match, they actually removed by an exonuclease, and ultimately this actually form a palindromic sequence and N nucleotide sequence. You can see here the introduction of P for palindromic sequence. This is generated by the Artemis enzyme, and this N nucleotide is added by TDT. So this 2 enzyme is very important for the generation of diversity. So the introduction of P and N nucleotides diversifies the joint between gene segments during immunoglobulin gene rearrangement. Immunoglobulin means antibody. So now these gaps are filled by DNA synthesis and ligations by the XRCC4, Sarnunas and DNA ligase 4. Then these actually D and J segments joined with each other, and 2 palindromic sequence and 1 N nucleotide sequence are actually synthesized. You can see here the combinatorial diversity, how this diversity is generated. VJJ recombination means to generate diverse set of antigen receptors. It could be antibody or T cell receptor. Now you can see there is a simple or example, like if the V sequence of heavy chain is 41 times, the D sequence is 23 times and the J sequence is 6 times, then the possible number of combinations of these three VJJ sequence can be 5658. Let's assume there is kappa chain that is associated with the heavy chain, and this kappa light chain consists of V segments of 41 and there is no D segment but J segment of 5. So possible number of combination here is 205, and the lambda chain can produce 165 number of possible combinations. But when we add this heavy chain, light chain together, we form another combinations in human that is approximately 2.09 into 10 to the power 6, a huge number of combinations. Just by this existence of different gene segments, we can provide a 10 to the power 6 number of antibody. But this is not only the reason behind the diversity. This is one of the reasons. There are 5 mechanisms that generate antibody diversity in naive B cells. The first is multiple gene segments, as I have already mentioned in the previous slide, that these gene segments like V, D, J can be in a different numbers, huge numbers, and in case of light chain VJ. So the first reason is multiple gene segment existent. These gene segments are present in many numbers. Then addition of P-nucleotides. When the Artemis actually generate P-nucleotides, means palindromic nucleotides, it actually add another diversity in the gene segment. And there is another function occurs that is called exonuclease trimming. This is the third reason for the generation of diversity. And the non-template N-nucleotide addition by TDT enzyme. And the last is combinatorial diversity.